Welcome to the Mechanisms of Developmental Patterning module. In this talk, we're going to focus on syncytial specification. We will first learn about the process of specification and the different types of specification. We will then look into depth into the concept of syncytial specification. The different stages that are involved that enable cells to determine their fate in the body include the stage of cell commitment. Now, cell commitment requires the process of specification to occur, followed by the process of determination. Once cell commitment has been finalized, then the cell undergoes differentiation, which allows the cell to develop specialized structure elements and distinct functional properties. Now let us look at each one of these stages. Specification is the first stage of commitment, where a cell is deciding what it's going to become in that multicellular organism. In order to understand specification, we have to go to the cells when they are in a blastula. In the blastula, the cells, which are the blastomeres, are not showing any characteristic properties of the cells that they will be developing to in the future. So if we take one cell from the blastula that we know is going to be a muscle cell further on in development, what we do is we take the cell and we put it in a neutral environment and look at what happens to that cell. If that cell becomes a muscle cell, then we say that that cell has undergone specification. Another example of that is, instead of using a cell that we know becomes a muscle cell, what if we took a cell from a blastula and when we culture it in a neutral environment, it becomes a neuron? Then in this scenario, that cell has undergone the process of specification because it has begun the process of becoming a neuron. Thus, in the case of specification, if a cell that is in a blastula is able to differentiate towards a given fate when it is in a neutral environment, then we say that specification has occurred. There are three main ways by which specification can occur. The first strategy is called autonomous specification. And in this scenario, the cell in the embryo has cytoplasmic determinants, which are usually in the form of mRNA and proteins that function to commit the cell towards a specific fate. Another type of specification is called conditional specification. And in this case, the cell in the embryo is able to commit to a specific fate based on cues that it gets from its environment. Thus, its fate depends on its position and interactions with the other neighboring cells and molecules that it comes in contact with. The third type of specification is called syncytial specification. And in this case, the cell fate is determined by the location of these cytoplasmic determinant factors like mRNA and proteins and the position of their nuclei in a structure called as a syncytium. The next type of specification and final type of specification we're going to learn about is syncytial specification, which is normally observed in insects. A syncytium is a cell that has many nuclei in the cytoplasm. In the case of insects like Drosophila, the zygote nucleus undergoes 13 rounds of cell division. However, the cytoplasm does not undergo division. Thus, you end up with a cell where you have a cytoplasm with many different nuclei. After the 13th mitotic cycle, the membranes are actually formed around the nuclei, and this process is called cellularization. The location of nuclei and cytoplasmic factors will determine the fate of these nuclei that were there in the syncytium. Now, the example of some cytoplasmic determination factors that will determine the fate of nuclei in the syncytium are bicoid and caudal.
Bicoid and caudal are found in the cytoplasm of the syncytium. However, their expression is not uniform. They form these gradients, and as we can see in the figure in the blue box, in the case of bicoid, we can find its expression to be quite high in the anterior part of the syncytium, and its concentration starts getting lesser and lesser as we move towards the posterior part of the syncytium. In the case of caudal, the opposite trend is observed. We have less caudal in the anterior part of the syncytium. However, as we move towards the posterior part, more and more caudal can be observed. Thus, these two cytoplasmic determination factors form gradients. The nuclei that are present in the syncytium, they will experience unique concentrations of bicoid and caudal depending on their position in the embryo. Now, bicoid and caudal activate different sets of genes that specify development of different body regions, as shown in the figure at the bottom. To look at it in more depth, let's go to the next slide. The nuclei that experience high concentrations of bicoid and low or no concentration of caudal eventually form the head, as shown in the figure to the right. The nuclei that experience low concentrations of bicoid and caudal eventually form the thorax of the insect. The nuclei that experience low or no concentrations of bicoid but high concentrations of caudal eventually form the abdomen. Thus, after cellularization, the cell fate is determined autonomously and conditionally. However, before cellularization, the nuclei already know what they're going to become through the process of syncytial specification.
With this, we come to the end of our talk, where we learned about the different types of specification and what happens in syncytial specification.